I tend to talk fast. I told Andre, and I'm telling everybody, slow me down. The concepts I'm going to be covering are pretty simple from my perspective, but I know there's some of you that don't know, they're not biologists and stuff, so slow me down, ask me a question, tell me if you're talking too fast, just, cool me down. So I'm going to be talking about the phytoplankton, just generally, give you the idea of why phytoplankton is important more from, ecological perspective, biological perspective. But while I'm doing so, I'm going to try to make connections back to the optics. And, what is it about phytoplankton that allows us that connection back into the optics? And you're going to see that the traits of the phytoplankton that are important for the carbon cycle and ecosystem are actually very similar traits to the one that we use in order to discriminate them using optics. So it's kind of pretty cool. But every time when you think about the tools that you can use to study optics, so for example, the stuff that I talked yesterday, when Colin was talking about absorption, and then he asked about how to convert absorption to the chlorophyll. That's important, but think about they're absorbing light. Why they're absorbing light? They're absorbing light for photosynthesis. Blah. And you think about, these optical measurements are actually not only optical measurements to tell you something about the quantities, but they're optical measurement of the capabilities to deal with light, and it's really important. Another note, this is a poster that an artist that was with us on the last cruise made, and it's really awesome. Awesome. She took imagery from Phaeocystis that you guys are going to be using on Friday and did these drawings. Taught us how to make drawings. I suck. So if you click on the link in your PDF that was uploaded, hopefully, you can download. It takes you to the PACE website where you can download this poster if you want to print it out. So this talk used to be given by Mary Jane, and she would ask everybody to write on a piece of paper what are phytoplankton. And I'm more of a yelling type, so I can ask you to tell me, what are phytoplankton? Anybody? Marine plants, okay. More? Okay. Nobody else? Has you guys heard of that?
Small organism that does photosynthesis and doesn't move, like it's driven by currents.
Okay. More? Single cell, small organism, small plants, move around, not by themselves. Second and third row doesn't have any opinion. Drifters. You don't, yeah. You've seen this one before. Last row does not count. First, second, and third row is asked for the input. Third row, come on. Any opinion on phytoplankton? No? Produce oxygen, awesome. They do what with carbon dioxide? They fix carbon dioxide. Great. Do they do anything else? You guys don't have any opinion about phytoplankton? Really? Have you heard about phytoplankton? Okay, cool. So what did you hear about it? Is it cool? Is it important? Why? More coffee tomorrow. All these things that your colleagues have said about phytoplankton are true, no? The first time when this term phytoplankton was coined was in the end of the 19th century based on, questionable resources online. And if you try to do the translation of the word itself, it actually does mean drifting plants. In that point in time, we were considering them to be plants, no? And one important thing when you think about the word phytoplankton, it does not explain a group of organisms. It's not like invertebrates, so, or like Lepidoptera, or like different families. This is a whole completely group of organisms that functions in that way. So kind of more, it's a word that explains a way of life. And what it says, that these are plants that drift around. At that point, we thought that these are plants, no, but now we know because of the rise of better microscopy and molecular techniques, they're not plants anymore. I'm going to cover that. And they are drifting from the perspective of the gigantic scale. Some of them do have capability of motility, but these are really small situations, so they can just move a little bit. Sometimes they can move up and down in the water column, but these are not really, they're not mobile with the water masses. And that's really kind of cool because we can use that in satellite oceanography to go back to the physics. So if we talk about, again, the fact that these are not just a certain group of organisms, when we talk about the phytoplankton, there's a great genetic diversity in there. It actually covers everything. I mean, Some people, because of the presence of certain pigments in archaea, might consider phytoplankton, archaea some phytoplankton, but I would stay away from there. But we definitely have presence of bacterial species in phytoplankton, have a presence of tons of different eukaryotes. So the cells, they have a true cell, the true organelles and everything, and type of organisms that don't have the true cell. They're more like just, bacteria, just a little bubble with everything inside. So really, really great genetic diversity, great taxonomic diversity. When we think about them from different trophic perspective, if we just talk about general plankton, there's so many different types. And this schematic is very protistan driven, so it's very driven from the side of true cells, prokaryotic cells. But we can see that it spans different size ranges, the ones that Colin was mentioning yesterday. And it spans different trophic levels. So when we talk about the phytoplankton, we talk about the plankton that is capable of doing photosynthesis. So these are the phototrophic phytoplankton. And you can find them in all different size ranges. But these can also be mixotrophic. What mixotrophic means that they're capable of fixing carbon dioxide, produce oxygen, make their own food, but also in certain situations they're going to go after organic sources of food and start munching on other things or try to get that carbon from different sources. You have heterotrophic species in plankton. They're not capable of photosynthesizing. And those are the ones where we actually enter in the region of zooplankton. And you have the whole suite of bacteria. Again, these bacteria can be also autotrophic, or they can use light, but they can use many different things. But here, this is just the general diversity of the plankton. So they're really diverse size-wise and capability-wise. Phyto, word comes from the fact that they thought they were plants. And to the furthest extent, they really have a great similarity to the plants because they can photosynthesize. The fact that they have a chlorophyll, this magical pigment that you heard about, that you're going to hear about for the rest of your life, allows them to photosynthesize. And phytoplankton, chlorophyll, as you're going to hear, is part of this really complex mechanism that allows for that. But to the first extent, the process of photosynthesis allows them to draw down carbon dioxide, same as plants do, to make carbohydrates, which makes them right away the base of most of the marine food web, and they can produce oxygen. So it's awesome, because if you think, by drawing down carbon dioxide, they're actually controlling the climate, They're making food, and they're making air that we breathe. So they're extremely important, extremely. Like, dolphins, if they disappear, they'll be really sad. It's really, really sad, and I'm sure there will be some shifts in ecosystems, but if these guys go away, we're dead. And just remember that. This process of photosynthesis can be simply written in this way, but it can be written more complex. But this is what's happening. Once again, carbon dioxide from the surrounding area with water, presence of light, very needed, help with chlorophyll and all the other complexes, make these carbohydrates and oxygen. And this is the basic of photosynthesis. I'm going to touch about photosynthesis a little bit more, but it's kind of like, I think Colin probably is going to touch upon it a little bit with the primary productivity, but this is the thing that you have to remember. And this is what makes them important. That's why we want to study them. That's why I want to study them. There's many other different reasons, but this is what makes the thing work. They come in many different shapes, colors and sizes, and internal structures and stuff. They come in many different types. The type that they have, their color, shape, and size will define the role that they play in the oceanic carbon cycle and ecosystem. Now, and you can see here just the imagery of many, many different types, predominantly prokaryotic. So this is a diatom, this is a silicoflagellate, this is a dinoflagellate, this is a teeny tiny cyanobacteria. So you can see they're really, really diverse. Shape, the sizes are not to scale at all. Doesn't matter, I just wanted you to see them. They actually come with many different colors. They have many different pigments. The coolest stuff about this is their shape, size, and color, and internal morphology and stuff will define their optical signal. So with understanding the type, you have this connection, you can make the connection between optics to the carbon cycle and ecosystems. Because these traits that phytoplankton have will determine their role on both sides. Both towards the physical properties, which we can measure with optics, and also the role that they play in ecosystem. So it's a pretty cool thing. So we really want to know a lot about them. We want to be able to study them. So when we talk about the phytoplankton, groups of phytoplankton, we can approach this from many different ways. We can talk about them from the perspective of particle. Just treat them as a particle. Forget the fact that they're kind of alive and talk about their size, their shape, the internal morphology. I'm going to touch a little bit on that one because we're going to cover it a lot on Friday during the particle size distribution class. Okay, Emmanuel's going to talk about physics. I'm going to talk a little bit about their capabilities. Colin's going to take it towards the particle size distribution regarding the phytoplankton functional types. So we're going to talk about it. I'll touch a bit about that stuff. They're going to talk about the taxonomic approach of discerning different types of phytoplankton. I'm going to talk a little bit about molecular methods, but also the old traditional methods that which names you heard about, and they're going to be repeated consistently throughout your life. The chemistry is really important aspects. We can look at them through the eyes of the chemicals. They have many different pigments. Most important one being chlorophyll, but this diversity of the pigments actually allows you to discern them in different groups. They have different minerals. If I go back, what you're seeing here actually visibly, it's mostly this mineral inclusion they have in their cells. So silicoflagellate has silica, no? Diatoms have silica. Where's the coccolithophore? The coccolithophore has calcium carbon. They have capabilities of having minerals inside of them. So that's a really important thing when it comes thinking about them as a chemical and thinking how they convert and change the pathways of these chemicals in the ocean, no? The capability of taking them off from the solution, make them particulate, and then take them deep in the ocean. Goes back to the carbon cycle, goes back to the ecosystem. And they can produce different metabolites, different chemicals. For example, toxins. So we can group them as harmful and non-harmful. We can group them as DMS producers or not. DMS is I'm not going to go to that, but it's a chemical they can produce and goes out in the atmosphere, actually starts changing the atmospheric conditions and actually initiating formation of the clouds. Emmanuel can talk about that stuff. He's on a cool proposal associated with that stuff. And then lastly is their function and role. Going back to that idea, the role that they play in ecosystem. You know, they have all these traits and that was going to impact the role that they play in ecosystem. And this is where the whole idea about the phytoplankton functional type comes from. I know that a couple of you are working on your PhD thesis looking at the phytoplankton functional type. And this is like a mix of all these things that allows them to define them as a phytoplankton functional type. And I'll go a little bit more into details what that actually means and how important that is and what can we do with it. Cool? I'm not talking too fast? Everybody can? Okay. I will speed up. So still stay attentive. Okay. So just from the size perspective, this was what Colin was talking about. She was talking about this basic dogma that we have about defining phytoplankton as particles. I have no idea who invented these stuff, but it's kind of cool because it goes in order of magnitude, no? So we said everything smaller than 0.2 be considered dissolved for now because, probably don't have capabilities of measuring, but we don't have really life existing here. Life, depends what you think about viruses, but like life. Then we enter in this realm of the picoplankton. This is where you're going to find lots of bacteria. Now you're going to find autotrophic bacteria, so bacteria that is capable of photosynthesizing, but you're also going to be finding heterotrophic bacteria. Although they're not capable of photosynthesizing, they will contribute to your optical signal. So you have to acknowledge these heterotrophs. One other thing that you're going to find here is that quite a lot of these, majority of these pico-prokaryotes, so picobacteria, are going to be nitrogen fixers. And this is where you're going to find them sitting. You're going to find them sitting in other places, but this is where the majority of them is. This is where your Synechococcus is and Prochlorococcus is. You're going to find lots of teeny tiny pico-eukaryotes. We just start discovering them because we didn't have the techniques to see them before, but there's tons of them. And they seem to be really, really important also for the open ocean environment. Then you enter in this realm of nanoplankton. So from 2 micrometers to 20 micrometers. You have phytocalcifiers. This is where your coccolithophores are going to be sitting most of the time. You're going to have lots of diatoms, so your teeny tiny diatoms. And not only calcifiers, but you're going to have cells that are really taxonomically, organisms that are taxonomically close to calcifiers that are capable of producing DMS. So those metabolites. So we're kind of bringing chemistry into here. Now we're going into the macro region. From 20 to 200, we're talking about usually diatoms. We're talking about dinoflagellates. We're talking about these big cells, silifiers and stuff. And then you can go to big stuff and really big stuff, no? And once you get from the 200 micrometer size range, you actually, the phytoplankton that you're going to encounter are going to be the phytoplankton that are capable of forming colonies in order to survive, in order to optimize their uptake of nutrients or whatever, predatorial relationships and stuff. So chains of diatoms, some goo, I don't know what to call them, goo colonies and things like that. And then really big stuff. In theory, sargassum is a really big stuff. It floats around. You can call it phytoplankton. I wouldn't, but there's things there. There's an additional way that you can divide this nano-pico range, and it also has to do something with the availability of the large-scale filters that you're going to be using in your life later. Some people call the the region between 0.2 and 2, ultra phytoplankton. And then they additionally divide this region between 2 and 20 to 2 to 5 micrometers region, and they call this pico. So just if you hear it, what you're going to find if you're going to be doing some kind of like big experiments where you're going to have to filter large amounts of water, it's going to be very hard to find 2 micron inline filter. So that's why you're happy about the presence of this thing, because then you can publish stuff, because there's a name for it, because there's a 5 micron filter. It's very easy to think about, I know pico, nano, micro, but what do those size ranges actually mean? You know, we dump them all together and we think about them as this like happy organismal thingies that exist in the same life and treat them in the same way. But if you're a pico, then length, something that is length of a whale is nano, and something that is size of a Statue of Liberty is micro. So when you start dumping them and thinking about them as the same group of organisms, start thinking about their size. So, you know, if there was a me size of Statue of Liberty, would I have the same capabilities of eating the food? Would the same food interest me? Would the same amount of light be enough for me? Would the physical environment around me be the same? Keep this in your head. You're a pico, and then there's a whale, and there's stuff. I don't know how this big. This is really, really big, no? But just think about this phytoplankton spans from the size of you to Statue of Liberty, and even larger, no? All right. So this is how much I'm going to say about particles. But just so you know, we have this organized way that we shape particles based on size. There's lots of questions. As Emmanuel, raised the point, what is the size? What is length? As you get into the larger cells, they become irregular. They can't stop being a ball. They start being elongated. They start making the shapes. What is the size? Think about that as well. And we're going to cover that additionally on Friday. Oh yeah? Good? Yeah. Take a breath. Let's continue. Taxonomy. I started as a phytoplankton taxonomist. I was in Croatia. I was spending 6 to 8 hours on a microscope per day. It was awesome. No, I knew everything about taxonomy and stuff, and then I went to my first big cruise. And there was a guy with flashing lights. Everything was flashing. I was like, what is that? And he's like, oh, it's called optics. What do you do with it? It's like, you study phytoplankton. I'm like, you don't have to microscope? It's like, yeah. I'm like, awesome. And there was a point where I left everything in Croatia, moved to US, study optics. But the knowledge that I have about taxonomy comes out to be really important now when I'm trying to build these different optical tools to study different phytoplankton types. And we have to acknowledge the hard work that these people are doing. More and more so, we're going now back and developing instruments that they have capabilities of looking at these things through the microscope, because there's so much history, so much knowledge about it. We really want to be able to see that, optically describe them, and then make this connection. So always be nice to the people who sit on a microscope. They're doing a hard job. So our concept of taxa has evolved over the time. And it was evolving because of our capabilities of observing these things. As I said, in the end of the 19th century, we thought phytoplankton is plants. But we know now they're really not plants per se. Plants exist in a completely different environment. There's some similarities, but we know that they're like actually bacteria and the protistans in there. Then as we start getting into the genetic methods, the tree of life start changing like crazy, and you're going to see more. So our definition of taxonomy that is present in phytoplankton is completely driven by the development of the methods. I have to put this paper inside of the Dropbox. I would really recommend it because it really talks about the techniques for quantifying phytoplankton diversity. It's a really cool paper and goes through review. Has really great tables that leads you to all the recent techniques, but all the old school techniques, allows you to discern phytoplankton. They really talk about this problem, how this taxonomy has been evolving especially over the last 50 years because of our new development in the techniques. So in theory, we have, 2 different systems. We have this modern system of algal classification that has been developed really like crazy within the last 20 years. It's mostly based on primarily different genetic methods, different omics methods that allows us to see the similarity and diversity within this group of organisms that float around in the ocean. And also development of a higher precise methods in ultrastructure electron microscopy, And I'm going to put probably this paper inside that's from the last year. It's a really cool paper and actually talks about all these molecular methods that allow you to discern different taxonomic type within the phytoplankton group. Then we have this classical system. The one is based on microscopy. The one that I'm talking about when I'm saying diatoms. or dinoflagellates, or flagellates. If I say flagellates, there's so many different taxonomic groups inside, but there's like ecological niche that they occupy and the way that they move kind of makes them together. So I'm going to touch upon this because this classical system is going to get you back to all the old papers. It's connected to classification that is based on morphological characteristics defined by light microscopy that we're going to be doing here, and they're actually really important for the optics as well. And I'm going to show you some electron microscopy, but also it's connected with pigmentation. Pigmentation changes the color. We measure color. Awesome. So once again, back to this tree of life, phytoplankton is not a group of organisms as a taxonomic structure. It is a way of life. Way of life. The many different eukaryotic species occupied, and also some bacterial one. So if we look into this phylogenetic tree, and this paper should be, it's not, it's in Dropbox, I'll put it inside, talks about really how the development of different molecular techniques has allowed this view of the phylogenetic tree, in this case eukaryotic, to change. And most of us, when we think about eukaryotes, we talk about, fungi, protistans, and animals. Actually, and then like within protistans, there's many different things, but this is like a Whittaker 1969 view of the things, and this has changed so much, tremendously. Every time a new technique has been built, so this is where we are now. This is our tree of eukaryotes now. The things that we're going to be looking at are going to be here, they're going to be here, they're going to be here, some of them are going to be here, some of them are going to be here. Animals are teeny tiny, and we're teeny tiny too. So this is the great diversity we're going to be encountering in phytoplankton, just so you know. When it comes to the bacteria, it becomes even more complex. I couldn't find a really good tree that captures all the autotrophic bacteria, but I just want to show you this. This is just a phylogenetic tree of the Prochlorococcus, and Prochlorococcus is one of these really important prokaryotic bacterial autotrophs. They're everywhere in the ocean. It just goes to show that if you're taking different methods here, using a specific portion of the RNA to do the taxonomy, and here you're using the whole genome tree, you get different groups. And that becomes a bit of a problem when you start organizing your data and so on. And we're not going to go too much into details, but I can tell you that it's bugging me a lot. So again, great diversity, different techniques yield different things, but there is a consensus that there's more groups building around this stuff to build like an overall consensus on what is the best way to go to understand the diversity from the genetics omics perspective. But the one that is kind of safer to run back to, especially if you're not an omics person, is the taxonomy one. No? And I'm just going to go through, not through different types of phytoplankton, but just the ones that you're going to be hearing about, the sexy ones, no? The ones that are important for class, and the things that are going to be rolling off our mouth and just have to understand. Again, when to say classification is based on morphological characteristics, mostly associated with the light microscopy. No? And there's some electron microscopy that's helping us discern different species, and we also use pigmentation. So here you can see a dinoflagellate and a pretty diatom. Okay, so I'm going to tell you what those are. Let's start with the teeny tiny one, Cyanobacteria. No? Those are the teeny tiny prokaryotes. It's a bacteria. No? They don't have internal organelles. What they have membranes, and they have things kind of connected to the membranes, and their DNA is really not put in any kind of bubble. It's just kind of like piled up in the center, no? So think about what that does from the optical perspective. You actually have, a little bowl with things inside. There's tons of different, cyanobacteria are like the most abundant phytoplankton in the ocean. We didn't know about them until like 20 years ago, maybe, when Penny Chisholm, by accident, it was Penny Chisholm, no? I think, yeah. By accident, started looking at the fluorescence and signaling, start seeing this million little dots. It's like, what is this? And this is how we discovered, pretty much by accident, cyanobacteria. And as we're studying them more, we're finding them everywhere in the ocean, highly abundant. But they're teeny tiny, okay? Many of them have capability to do nitrogen fixation. Who knows what nitrogen fixation is? Can somebody tell us a bit? Awesome. Nitrogen fixer. Go. From the surrounding water. Yeah, from wherever you are. So in order to grow, phytoplankton needs, everybody had a plant in their life. They probably killed, but you know how you have to give some yum-yums to the plant, and that's why you give them, like, you know, fertilizers and stuff. And what is in the fertilizer? You have nitrogen, you have phosphate, you have iron, and so on. So similar to the plants, the phytoplankton has their nutritional needs, and there's many different nutrients, as we call them, that they need. But one of the most important one is nitrogen. And if you think about it, you know, if you learned a little bit about biochemistry of the cell and just molecular concepts of the life, nitrogen is in lots of things, no? There's some in DNA, and some in proteins. It's really important for the proteins and so on. So you need that nitrogen, you know, and many other things. So what these guys do, for different than most of the other phytoplankton that's relying on the dissolved nitrogen in the form of nitrate, ammonia, urea, these guys can exist in an area where there's no nitrogen inside of the water and actually go after this gas that is available to them, and they do this nitrogen fixation. Now, one cool stuff, this is a cool part of the story, is that the enzymes, they can do nitrogen fixation, cannot exist in oxygenated environment, no? So what do you do now? You're phytoplankton, you're producing oxygen, no? But your nitrogen thingy enzyme cannot work while oxygen is present there. So it's really cool to actually go, and I would urge you to read if you're bored terribly, different ways that they have developed. These single cell prokaryotes, they develop different ways of kind of either temporally offset stuff, so they take down, they do the nitrogen fixation during the night and oxygen production during the day, or they actually form a specific cell in which the nitrogen fixation happens, and then they kind of like form a colony and talk to each other and like, hey, I'll give you some oxygen if you give me some nitrate. It's pretty cool. So it's you know, when you think about this capabilities of the single cell prokaryotic organism they can do, is really cool. You know? They're most abundant phytoplankton in the ocean. Keep on thinking about that stuff. They're not visible, they're not sexy, they look like balls. There's nothing cool, sexy looking about this stuff, but they're extremely important. They're present everywhere. In tropical ocean, cosmopolitan ocean, so on. One really cool one that you can see with your eyes is trichodesmium. Okay, trichodesmium, and Australians and tropical people will know, a trichodesmium is actually a colony. There's many different cells piled up together, and there's so many of them piled up together, they make actually things that you can see with your eyes. You can actually capture it on a filter and see these little puffs and buffs. But then they can form these gigantic blooms. Here, I'm going to make these puffs and buffs, and they can make gigantic blooms that you can actually see from space, no? So this is a trichodesmium bloom, this is a trichodesmium slick. There's like this like glue, this slick that happens on the surface of the ocean. And they're superbly important because they bring the nitrogen in the system, because the nitrogen that they fix now, after they use it, when they start dying, comes back into the system, so it feeds the rest of the population. Okay, so just fast about the other types of cyanobacteria. The words that you're going to hear is Prochlorococcus and Synechococcus, teeny tiny round balls, no? Extremely important. If you look at the distribution, Prochlorococcus is going to be linked kind of to the subarctic, no, from the subarctic south, so equator up to like 50 north and 50 south, really abundant, really superbly important to support this like very oligotrophic parts of the ocean. On the other side, Synechococcus you're going to find everywhere. They're really not competing for the same niche. They can exist in the same space and time. Honestly, under the microscope you can't see them except for the dots. But what we can see, we can use diagnostic pigments to discern them, and ultrastructural microscopy usually. So use this technique called flow cytometry to actually see them in your samples. Go ahead.
They're not seasonal. Like I know for that, so Coccolithophores are very abundant in the summer, and the Synechococcus like.
And that's up in where is the
In the Red Sea.
in the Red Sea?
Close a Gulf of Mexico.
I mean, there will be seasonality. There was a really cool paper published by Heidi Sosik, I think, a year ago. He's actually looking at the seasonality of the bloom of Synechococcus offshore New England, and she's seeing some shifts in potentially climate-driven. So there is a seasonality to their presence. I'm just not talking about the seasons and stuff here. I'm just talking about the presence of absence, and understandably so. There should be a seasonality. They're plants, no? They have their springs, they have their plumes and stuff. And yeah, that's not the aspect that I talked about, but there is a seasonality, yeah. Trichodesmium, also appearing seasonally. If you want to see them, you want to go around Fiji in 2019 on a cruise with me. help me filter, and actually start looking at these slicks that appear because of their capability, as I said, to form these colonies and mushes and so on. They're really cool and they're really studied a lot, really much better than the rest of the cyanobacteria just because they're visible. We knew about them for a long time, you know? Because of them being nitrogen fixers, the other one, they're very sensitive to the presence of phosphate, especially presence of iron. Iron is needed for the enzyme that helps them fix the nitrogen. So they're going to be found in the areas where there's iron and there's at least some phosphate. This is probably taken in the around January, February, southern hemisphere somewhere. I should have looked up tropical, Fiji-ish, hopefully. But you can see them from space. They sometimes make these patches. They're like 100 kilometers big or even bigger. So, you know, this is 20 kilometers. So everything here all the way up is a trickle bloom. So this would be, what, 200 kilometer? Gigantic. Cyanobacteria, bacteria from space. Awesome.
So is there a satellite product that
I am familiar with several different papers that have been published on looking at trichodesmium from space. And I can share those papers with you. There's not a standardized NASA product that will give you a trichodesmium. There's actually a really good review done by Lachlan McKenna, her co-advisor, that has done the review of different trichodesmium algorithms that you can use to see them from space. With NASA or any other space agency, as far as I know, we don't have a standardized product that can give you a trichodesmium output. Cool? Is that a good diplomatic answer?
yeah, I mean, there's a follow-up question I get.
No, go ahead.
What does it take for NASA to accept or to standardize an algorithm?
We're going to take most of the algorithms. If you have an algorithm that you want us to look it up, we can talk to us. We're going to take it, put it in a code, and put it in as an experimental algorithm. You know, for it to be fully operational and stuff, I don't know. I think there is some standardized process that you can ask Jeremy about in the third week. So Jeremy and Sean would be the responsible one. I would assume it has to do with science and a bottle of scotch. Probably mostly science, and then, you know, a bottle of scotch would help out. But I think there's a standardized, you know, a series of things that have to be satisfied in order for things to be operational algorithms, standardized operational algorithm. We have tons of, if you go on our website, you're going to see tons of experimental things. So whoever comes up with the idea and is willing to work with us, we're going to take their code, put it in a system, and produce these products as experimental ones. Definitely. Go ahead.
When they talk about atmospheric correction, is that trigo tends to stay right at the surface and tends to screw up the atmosphere.
Exactly.
It's pretty bright in the red.
Come with me.
Water's dark.
Exactly.
So there's another issue.
Yeah. And hence come with me in Fiji in 2000. I need a postdoc. I was counting on Niels, but he's not graduating yet. No, and that's something that we're going to look at. You know, the fact that they form these slicks. How does that impact the actual, like, you know, how does that impact the subpixel variability, the whole impact on the signal that is coming from the ocean, but also the fact that the atmospheric correction gets completely messed up because they do this infrared jump. That's what Lachlan does. All right. The next group is very complex to say. Primidozeophytes, you know? These are actually Coccolithophores and Phaeocystis. You know? It's a group that brings them together, taxonomic group, and there's lots of similarities between two of them. But everybody knows a lot about Coccolithophores. Coccolithophores are awesome. This is a Coccolithophore now. This is a Coccolithophore, probably. Originally used to be a Coccolithophore. So what happened, and when we started developing chalk, we actually went and digged for this calcium carbonate sediments, and we start cutting them up, and we start making chalk out of them. This is where they came from. Because calcium carbonate is taken, it's made in the shape of these shells the Coccolithophores have in them, and when they died, a long time ago in geology, started settling on the bottom of the ocean, the White Cliffs of Dover came up, somebody came, look, I can use it as a chalk, and chalk came up to be. Go ahead.
Are all Coccolithophore calcifiers?
No, I'm going to get to that. Thank you.
Uh-huh.
Yes.
Do we know how these plates affects the light absorption of the
Who did the plates?
The plates, the cup.
They terribly, yeah. There's quite a lot of work being done. I'm going to touch upon that. Yeah. So not all the Coccolithophores are calcifiers. And some Coccolithophores that are calcifiers will be calcifiers only in specific environmental conditions. So, but when we think about Coccolithophores, we think about the calcifiers. Because many of them, they're not. But thankfully, the ones that are calcifiers, we know more about them just because the fact that they're calcifying, making these coccoliths, these plates in them, helps us, see them better, know taxonomically more about them. If you think about size ranges, this Coccolithophore is somewhere around 7 nanometers, no more. 7 to 10 nanometers long, no? So this is its length, no? So each of these plates then would be 1 nanometer long. Each of these, so this is 1 nanometer, this would be a third of a nanometer. Sorry, micrometers. So like 5 micrometers, 10 micrometers, 1 micrometer, a third of a micrometer. How much is a third of a micrometer? 0.3 micrometers. How much nanometers is that? Milli, micro, nano, pico. Milli, micro, nano, pico. Milli, macro. Milli, micro, nano, picos. 10 to the third every time you do it. You remember the song, milli, 10 to the third. Micro, 10 to the third. Nano, 10 to the third, pico. So 0.3 micrometers. How many nanometers is that? Bingo. What are the wavelengths that we're looking at? 400 to 750, no? So just to give you again the scale of these structures, no? For the types of Coccolithophores that contain these plates on them, the mineral structure in them interfere with light in such a way that they scatter like crazy, and that has to do something with the chemical composition, the mineral type, but also as well as the morphological structure about this. I think we're going to talk about a little bit more on Friday about these, how much internal morphology and shape modifies that. But they're really, really big scatterers. And that's one of the reasons why we see this Coccolithophore, this calcium forming, coccolith forming Coccolithophore is easy from space. I'm going to show you an image. But again, as Emmanuel said, not all of them make calcium shells, and not all of them make calcium shells all the time. Go ahead.
Does something happen with scattering when the wavelength light approaches the size of
I'm going to talk about that. I just wanted you to remember that. I'm talking taxonomy, and I'm just throwing things. Keep that in mind for tomorrow. Tomorrow? Yeah, tomorrow. They're very sensitive to the temperature. They're very sensitive to the acidification. Capability of actually precipitating calcium carbonate, making this calcium carbonate from the surrounding ocean is very dependent on availability of the calcium, and which is driven by the changes in the pH can change its capabilities of making calcium carbonate shells a lot. So many studies, you're going to see many studies looking at the calcification rates of Coccolithophores that have capability of making these calcifiers, as a function of this changing pH of the ocean, ocean acidification. They go up and down. No, there's not like, oh yeah, all the Coccolithophores will die. Some of them will not. They're extremely important for the carbon export, the ones that make coccoliths, because when you think about export carbon, when we talk about the carbon export, we're talking about mostly organic carbon. But this is also carbon too, it's calcium carbonate, no? So this mineral ballasting, they take when they dive and start sinking, they take also carbon, the one that they precipitated or down in the deep ocean with them. So they're really, really good and important to take in consideration when you're doing some kind of models or estimating carbon export. Phaeocystis is one of these promisiophytes that kind of falls in a different group, and it's really important and very abundant in world's ocean, especially in Nordic areas and stuff. It's a colony. It's a bunch of cells embedded in this goo. I don't know how else to explain it. It's a polysaccharide gel matrix, yeah, but it's a goo, no? We know about them a lot because British wrote about them, about the smell of the shoreside. There's many poems how in the British went on their stroll on the shoreside and it smells like an ocean and stuff. Which actually smelling is this DMSP, which is one of those metabolites they form. when it tends to bloom, it tends to be very ugly looking because this polysaccharide gel matrix tends to capture the bubbles, and then you make these big foams on the beaches, and you're going to see the photos if you Google it, of this foams actually entering to the towns and rolling through the towns because it just gets accumulated. It's it's an organic. Put some goo, organic goo in the water and make bubbles. They're going to be pretty stable. That's what happens to them. Once again, they're also important for the carbon flux in the ocean. So this is this Perminiziophytes, no? And once again, we're really like Coccolithophores. I think at least the Coccolithophores, they're making calcium carbonate shells, or the one that have capability, because the fact that they interact with light, because of their mineral structure and the shapes they have, they scatter light in unbelievable ways. As you're going to later hear, that their capability is actually backscatter is really great. So we can see them from space. As Colin was saying yesterday, it changes color to milky. Ken was saying that, Gulf of Maine becomes turquoise or something. And recently there was a whole article about Black Sea turning turquoise. It was in the New York Times, no? And actually, the person who spoke to New York Times, Barney Balch, who studies these Coccolithophores, is going to be here on fourth week and talking to you about his awesome work that he has done. In order to develop his algorithm, at one point, during what was the name of the experiment when they dumped tons of chalk X? Yeah, it was a chalk X. They took tons of chalk here in Gulf of Maine and dumped it. And it's amazing. Everything was white. That ship will never, I think, Endeavour will never allow him to go back on because everything was white. Everything. There was an Indian postdoc of his. It was really funny. There was, it was the only time that Joachim was just was like, oh, it was it was so funny. Anyways, if you ever see these photos, ask about, if you see ever Barney or Joachim or your Endeavour, ask about chalk X. Everybody's going to start pulling out the photos. What they did, they actually dumped tons of chalk, certain concentrations of chalk, and because they knew that they could see it from space, they were using take that to base their algorithm development. Luckily, they did it in Gulf of Maine, where it's really cloudy, but this is a different problem. So Dinoflagellate, this is another group that's going to be rolling off our mouth, and these are the relatively big, not always, but relatively big cells. And we know we're mostly looking at these big cells, but they exist in nanoformats too. They're the algae that have these flagella, two flagellas that allows them to travel. And one thing that I really always find cool to remember is that example of this species, for example, this one that's a serratium, they have two flagella. For difference than animals, where the tail goes after the puppy, they're following their flagella, no? So remember that. There's going to be one like here, but one flagella, they're following the flagella, if you think about their motion. That's one thing that is always. So they're flagella-containing algae. They're capable of swimming. Can they fight the big currents? Not really. We see them sometimes going up and down in the water column. We see them sometimes accumulating and kind of acting like as a kind of swarming behavior, especially the ones that are more on the heterotrophic side. So Noctiluca, if you ever find a little bay that there's Noctiluca, so it's with this one type of the dinoflagellate that has the bioluminescence properties. If you throw a rock at them, wait a little bit and throw the rock in the same spot, they're actually going to move. And we see them going up and down in the water column, and they see them really reacting to the different levels of turbulence in coastal environment. So they have this magical thing with emotion. Most of them have heterotrophic capabilities. So they're looking for the source of carbon in a different way, not through photosynthesis. But quite a lot of them have autotrophic capabilities. So majority of the dinoflagellates are actually mixotrophs. You're going to find them everywhere. But predominantly, the larger ones, the more important ones, you're going to find in coastal warm waters. You're going to find them as symbionts. So I have already, I don't remember last night, put a paper that just got published yesterday that talks about Zooxanthellae. Zooxanthellae are a type of dinoflagellates in that group that lives in a symbiosis with the coral. And it's really cool. This coral is capturing the blue light that is existing in that environment because it's deep and fluorescing it back as red. So the algae they're living deeper in their tissue could actually have a full spectrum for photosynthesis. So I'm going to put this on this paper. So these are these little guys that live in there are also full in this group of dinoflagellates. And they're red-tide organisms and sometimes toxic. So Gulf of Maine, Alexandrium, boo, evil thing, does not have to exist in large numbers, but produces really, really bad toxin. That's your heterotrophic dinoflagellate. The one that I spent my thesis on is called Lingulodynium. The one does not make toxin per se, but because it blooms in such a large concentration, when it dies and falls down in the bottom of the coastal ocean, what happens? What do you get when you throw a bunch of organics on the bottom of the ocean? Decomposition. Who does the decomposition? Bacteria. What does bacteria need? What do you need to live? Oxygen. What happens? Anoxia, hypoxia, anoxia, fishes die, everybody's sad, everybody's concerned, they give you money to do a PhD. So in California, no, I mean, it's more of a concern from human perspective, no? So when you talk about harmful algal blooms, it doesn't have to be that they only produce toxins. There's different other ways that they can harm the environment from the perspective of humans. So in this case, this is chlorophyll concentration of 160. Your average open ocean concentration is 0.01. This was thick. I mean, this is, California dreaming. It's blue ocean. This is how it was orange. So what happens when these guys die, they actually kind of get entrained in these little marinas and stuff. They die on the bottom, anoxia, hypoxia, anoxia, fishies die, and there's a big problem. So that's what I was doing. I was studying this one. So it does not have to be they produce toxins. It's just they're harmful towards the environment. Diatoms. Yes, go ahead. Yeah. Andre, no. So, if they have, so think about it. In order to all these different toxic algae, they can be diatoms. There's lots of toxic algae that are diatoms. I'm going to be talking about them. There are lots of toxic algae that are cyanobacteria. There are lots of toxic algae that are dinoflagellates. And they have different, as I told you, cyanobacteria is a teeny tiny, round, kind of not sexy. Dinoflagellates can be like really nice shaped forms, can turn things red. Diatoms tend not to have a specific color, but they can be traced differently. So there's so many different types of harmful algal blooms that it would be hard to make an optical signal that discerns them. So you really have to go after a certain group or certain type. There are different algorithms out there. There's for example, a species called Carenia brevis. I think it's Carenia brevis. It has a specific pigment. It allows you to trace this pigment and actually discern it, both from remote sensing and in situ. I know for Andre, I'm part of this project, yeah. So what they're doing is, because Alexandrium does not form so much large numbers, it doesn't have like highly and specific signal, you can actually go after the different water types. To try to discern different water types that that specifically harbor the existence of those algae. So when you're going after harmful algal blooms, you really have to go after different species. And Colin can say more so.
And the both named the toxic dinoflagellate never, which means like it's just
Yeah. And yeah.
But it does tend to co-vary with all the other dinoflagellates. So if you can detect when
Yeah.
all the other dinoflagellates come to town, then you know with probably 90% confidence that and it will be at levels that are very toxic.
Yeah, but you're not going to be able, so that's the thing, to find a water type. And think about it, what I said, dinoflagellates are usually big. What do Collins say about big species regarding absorption? Big versus small. What was the difference? Sorry?
Packaging effect.
Packaging effect. So the slope of absorption. So you can play with that. That would take you to discern Alexandrium. You can't be 100% sure, but you can say there's a big possibility of Alexandrium happening there. And you take some knowledge that you have from environmental conditions. There's lots of paper, and you can build like a nice algorithm. There's not a big magical patch. Evil, good. No. But they just. So think about all these things, their environmental conditions. When you think about developing algorithms, don't think about just about their optical signal. Going back to the stuff that I said, the same things that define their traits will define their role in carbon cycle and ecosystem. And also they will define their optical signals. So try to use the ecology that you know about that species or something specific other than you might be able to, use a vector to discern them optically. Diatoms, okay. Diatoms, and we're going to do 5 seconds break, okay? Diatoms, the most beautiful ones. I love diatoms. Diatoms are gorgeous. I mean, look at them. They look like you want to make jewelry out of them, no? These are the great images from Christian Sardet, and I would definitely recommend his book called The Plankton Drifters. It's just a book that gives you beautiful images of plankton and also tells you a little bit about the taxonomy and their ecology. And so he was part of the project Tara that Emmanuel was one of the main guys on. He brought this beautiful, gigantic microscope that I tried to turn on and never succeeded. But he took gazillion, these gorgeous images. And if you go online and actually find a video containing this, sorry,
They call Plankton Chronicles.
Plankton Chronicles, yeah, the videos.
There's
Go,
beautiful movies.
and you wanna find this movie. If you're gonna take a break, I might find this movie. I'm gonna find this movie during the break. We're gonna take a couple of minutes break after I'm done with Diatoms. We'll find this movie and show you something and ask you a trick question. Then I have a big NASA sticker for whoever knows the answer, okay? Okay, so I'll just talk about Diatoms. They're really cool, they're really important. One thing that's specific for them, usually that you find them in nano size, micro size. They have silica, okay? So the silicifier, they take the silica, they need silica to grow. So that defines their ecological distribution and stuff. In the areas where you don't have silica, you will not find diatoms, no? They need diatoms, they need silica to grow. They put silica into their shells, and when they die, not only they take this carbon, organic carbon with it, but also take silica in the deep ocean. That makes them kind of effective sinkers in a way. There's two different types of diatoms, and here I put both of them being centric. But they can be centric. That means in cross-sectional area they look, I'm going to take your cap like this. They pretty much look like this, most of the centric ones. What you're seeing here is a bunch of them together now. This is also a centric one. It's kind of a little more like a pill shape, but it's a centric one. Then you can have pennate one. I have some images. Pennate one are kind of like longer. They don't have, they still look like a pillbox, but they're kind of longer, have elongated shapes. They usually do not form stacked up chains. They'll make elongated chains. Pseudonychia, you might heard about it, the toxic algae. That's one of them. There's some of them are toxic. Pseudonychia, the favorite one. You know, everybody knows about Pseudonychia. Destroyed the west coast for the last couple of years, and it was afterwards connected. I think there's a paper by Ralph Gudella with that blob that was sitting up in Arctic and region of Alaska and stuff. The hot blob changed the circulation, changed the environmental conditions in such a way that Pseudonychia was blooming. So if you want to go capture Pseudonychia, it does not have an actual optical signal. But again, think about the ecosystem, the ecological niche that it occupies. So if you can define the water type, the environmental conditions of a Pseudonychia, you can't say it's there, but you can say, oh, with the probability using remote sensing, there's a big probability it's there. And they can also be a good tool to discern and, you know, alert people for the harmful algal blooms. The spring bloomers, they're the creatures of the spring. They come first. You know, like when you see this image in a salad, you know the spring is there because it's blooming in North Atlantic. It's yay. And they're effective carbon exporters. They're really important for the carbon export in the open ocean. Most of the time, what we really want to know to the first extent is to differentiate the chlorophyll that is associated with diatoms in the open ocean versus other ones, because they can allow us to really decrease the uncertainties in carbon export from space, because these guys are really effective carbon exporters. Go ahead.
All the species of the phytoplankton grow in spring, in spring the temperature medium and the sunlight is very, make a very phytoplankton. Do all the species grow in spring or in different time?
Okay, so what happens in the springtime? Let's think about what happens in the springtime in the ocean. No? What happens in the springtime outside? What are the changes that define spring? You have increase, more light. If you're Maine, the floor, the mud is the mud is coming. So there's this like temperature and light associated changes. So what you get in the ocean is during the winter it was really cold. No? And I'm talking about North. I'm going to position myself in North Atlantic. It's really cold, and just because of the coldness, from the surface towards deeper areas of the ocean, there was no stratification. The difference in the density of the water, because the coldness is usually because the wind starts disappearing, and you start getting this mixing. The mixing, what it brings deep water up to the surface. What was in the deep water? What was I saying? Things when they die, they go down, no? They start decomposing and stuff like this. All this yum-yums, all this nitrogen fixed and dead silica and stuff like this now got dissolved thanks to bacteria, thanks to chemical properties coming back to the surface, no? And it's your winter. But if you're mixing this water deep and you're phytoplankton, you're now on 0, and then you get it mixed to 700 meters, then you're back on 0 meters, 700 meters. What happens at 700 meters? How much light do you have? 0, exactly. So most of your time you're going to be spending and not really illuminated portion of the water column. No, so you're gonna have sunlight to grow, but not really to make bloom. And okay, disclosure here, I'm talking about classical Swerdger bloom here. Okay, just so everybody knows. What happens in the springtime, because of the different physical processes, and we can discuss them, is you get formation of the stratified water column. It can be because of the temperature. So the temperature, the surface of the sun is out there, the days are longer, you actually start heating up the surface, and the surface starts getting heated up. It's getting heated up, the temperature's changing, it's making changes in density. So the surface area becomes different density than the water underneath. So then what you have is this physically separated portion of the surface of the water now has yum-yums from the winter, has light, because now they're just like phytoplankton, just beginning mixing this teeny tiny portion of the water. This is stratification, and this is where the bloom starts. So who's going to start blooming? The one that can take stuff first. Okay, the one that can divide first, the one that has the most capability of taking the nutrients first, the one that doesn't have any enemies. In theory, everybody's going to start trying to bloom. But the teeny tiny guys are going to have teeny tiny enemies. They eat them really fast. These guys have a capability of taking nutrients really, really fast. There's the reason why we see them winning in the open ocean. In coastal area, it doesn't have to be that way. Usually this limitation of the silica, no presence of the silica, that's going to throw away the diatoms, and you might have somebody else blooming first in spring. What I forgot to say, for example, solstice, that's where these guys are blooming. Pretty much when you see solstice, you're going to start seeing things in New York Times, like, oh, look, here, there's Coccolithophore bloom. Go through the newspapers, it's always going to be around solstice. So there's a seasonality to all of them. But these guys are considered the bearers of the spring. So I'm going to take a 5-minute break here. Okay, I have a little bit of talk about pigments and function role, stretch out, pee, go smoke cigarette, whatever. I'll try to find that cool video, and then somebody who ever gets a good answer that I'm going to have to the question gets a big NASA sticker, big one, so you can put it on your instrument or whatever. Okay, cool? Go stretch. Stretch now. Oxygen. Enjoy that beautiful oxygen that was produced by phytoplankton for you. I can give you my NASA spiel. I've been trained by the media. That's the best talk that I can give.
When we talk about the size classes, so let's say 2, 20, whatever. I know the particle distribution is not. I didn't ask that question now because I'm pretty sure it's going to be Friday, but it's not a log. It's not a normal distribution. And
Phytoplankton size distribution, particle size distribution. There's a difference. And plus, like if you think about, when you're talking about this, particle size, you're doing on a log scale. So this is the frequency, and this is your size. You can always going to have here. In theory, nature always tries to be in this line.
But that's a log.
It's a log.
Okay.
This is a log. It strives to be this way. It wants to be that way, but the only way that the nature can function if it oscillates around this line. So what you're going to have when you have a spring bloom, for example, your particle size distribution is going to be like this, because your diatoms are blooming. And 3 days later, it's going to be like this. The only time you're going to find this is a steady state situation, which doesn't really not exist a lot in the ocean. But nature strives to be here. Everything evolved around this line. But very often in a bloom situations, very transient environments, you're going to find, offsets of this PSD line. They're going to try to go back to its way by, predatorial aspects and so on. So is this a good answer to your question?
Yeah.
Or not?
No, it does, but not within these size classes, because we divide in size classes because, we can separate, because we have filters for that. That's pretty much it. So I use size classes in my master degree, but I did the whole range of size in the microscope. I measure every particle. And for example, between 2 and 20, there's at least 2 or 3 modes of,
Oh, yeah.
of distribution.
Oh, yeah.
And sometimes, for example, for my microscope, I was in the 5 and I couldn't go any further. And then we have another instrument, we have like flow
Yeah,
cytometer.
photometer, yeah, and you have that stuff.
And
there.
then we are still having trouble to like,
Define that small region, yeah.
yeah, the range between these two because the